Welcome to Chapter 10, Classification of Microbes. This chapter fulfills Objectives 2 and 4. Specifically in Objective 2, it's understanding the fundamentals of microbiology, and in Objective number 4, gaining an understanding of the interactions and the impact of microorganisms on humans and the environment. In this chapter, we're going to be looking at the study of phylogenetic relationships and classification of organisms. This is going to be revisiting information that we took a broad overlook at in Chapter 1. And also, we're going to be looking at methods of classifying and identifying microbes. Under the study of phylogenetic relationships, we're going to talk about the three domains and phylogenetic hierarchy. Under the category of three domains, you may remember from Chapter 1 that those three domains are bacteria and archaea, both of which are prokaryotic. And what makes a prokaryote different from a eukaryote? That's right, no nucleus. Now, from these three domains, bacteria, archaea, and eukaryote, we split into the other kingdoms and species and genus that we are more familiar with. Now, one thing I want to remind you of, over here in eukarya, we have fungi, animals, and plants. These are the multicellular organisms that we usually think about when we think about life. But you look at the rest of this chart, and we are vastly outnumbered. Now, archaea is smaller, and that's simply because we're still working on classifying them. Let's look at relatedness. If you look at how this tree is set up, this original point shows the lineage of the surviving cell. Now, in the beginning, there may have been more than one cell that evolved, but from the DNA work, we all have the same DNA code, whether you're a bacterium, an archaea, a yeast, a human. So this implies that all life descended from one organism that had one genetic code. And from there, bacteria split off early from the others, and then archaea split off, so archaea are more closely related as far as the genetics to eukarya. Now, eukarya is kind of a hybrid beastie because of our mitochondria. We are also related to bacteria, and because of the chloroplasts in plants, they're also related to bacteria. It's kind of like a marriage. We've got in-laws. Now on to classification of all of these organisms. We're going to talk briefly about scientific nomenclature, just to remind you of how it's set up. Then we're going to talk about taxonomic hierarchy. Then we're going to talk about classification of eukaryotes, classification of prokaryotes, and classification of viruses. On the subject of scientific nomenclature, remember that we have the binomial naming system. You have a genus name and you have a species name or a species epithet. So when you see the name of an organism, you're always going to see two names. First one capitalized, second one not capitalized, both italicized because it's based on Latin. And when you see a situation like this, where you see another genus name in parentheses in the center, this is because as we continue to do DNA research, we reorganize the various classifications. So in this situation, this particular organism used to be in the genus Pseudomonas, but as we did more research, turns out that it's not terribly related to the other pseudomonads. So a new genus was created, Burkholderia, and it was given a new genus. But because it was referred to in scientific literature with the genus Pseudomonas, they include that so that you can look back and find research with the previous name. It's kind of like a woman with her maiden name. So it's good to include the maiden name so that you can find that person that you went to high school with on Facebook. Now a reminder of how taxonomic hierarchy is set up. We always start with the domain. So it's either bacteria, archaea, or eukarya. Then we go to kingdom. Now, bacteria and archaea so far have the kingdom, just the one kingdom that has the same name. Eukarya has several kingdoms. Okay, and for our lovely example over here, the kingdom that we're going to talk about is fungi. So then from there we go to phylum. Then from there we go to class. Then from there we go to order. Then we go to family. And now we're going to get to what we were familiar with, genus, and then species. So this is the setup that we have in the classification of eukaryotes. We go from the domain, kingdom, phylum, class, order, family, genus, species. Okay? And we have the four kingdoms. Protista, and what makes a protista is that it has to be eukaryotic, single-celled, and not have a true cell wall. Okay? Then for kingdom fungi, to be a fungi, you have to be eukaryotic. You can be multicellular or single-cellular, and you have to have a cell wall that includes chitin in the cell wall. For kingdom plantae, now we've since moved algae in with the plants. Okay, so you can be single-celled, clear up to multicellular. You've got to have some sort of photosynthetic capabilities, chloroplasts in other words, and you, generally speaking, have a cell wall. Now, because we've included algae in with the plants, that cell wall can be made of just about anything, but the one that I want you to remember is cellulose. And then there's kingdom Animalia, which we are a member of. All animals are multicellular, eukaryotic, and we don't have a cell wall. After that, anything goes. Now with eukaryotes, when we are defining relatedness, it's a little easier because with the exception of Protista, we can tell a species because they can interbreed. Generally speaking, that's the most common definition of a species, are populations of individuals that can interbreed and produce viable offspring. When you get to prokaryotes, it's a little more interesting, simply because they don't engage in sex. So how do you define what a species is? Well, what we've defined a prokaryotic species as is a group of cells with similar characteristics, physical and biochemical, that show very, very close genetic relatedness. Now, a clonal population of that species is a population that arose from one cell. This happens in the lab. When we talk about a pure culture, what is it? It's a clonal population. It arose from one cell. We don't deal with this in animals and plants very much. Well, in plants, yeah, we do. But that's what a prokaryotic species is, whether it's bacterial or archaeal. Now, the place that we go to record all of our information about the various prokaryotic species is Berge's Manual. It's a wonderful manual. If you continue on in microbiology, you will become very familiar with Berge's Manual. So I wanted to introduce it. It's where you can find just about any bacterial species that has been characterized, and you can look up its characteristics, what to feed it on, what it ferments, what its GC content is, far more information than you would probably want to know. But it makes it very useful for somebody like me when we were taking samples from the periodontal pockets, that's the space between the gum and the tooth, of patients and trying to figure out what microbes were growing there. Berge's Manual is very helpful in identifying the various species. Now, when we get to viruses, it's even more tricky, because not only do they not engage in sexual reproduction, they don't even reproduce by themselves. And so we're still trying to decide whether they're alive, but they have genetic information and there is relatedness shown between them. So a viral species is defined as a group of viruses that have the same shape, same morphology, the same genetic composition, either DNA or RNA, and they occupy a similar ecological niche. In other words, bacterial viruses don't infect eukaryotes, and eukaryotic viruses don't infect prokaryotes, etc., etc. So that's how we classify viruses. So with viruses also, we have families, we have genus, we have species. That's as far as it goes with them. Now before we go on, let me explain why we do all of these different things that I'm going to be telling you about. Why we do all these things to identify microbes. As you've probably been able to tell from looking through the microscope at various organisms, you can't tell a whole lot from looking at microbes through a microscope. You can't tell a whole lot about them. They all look the same. They all look like little spots under high magnification. So we have to use different methods of identifying and classifying microbes than we would in, say, identifying and classifying bird species, in which you can do feather color and beak shape and color of the feet and various things, or plant species where you can say, okay, this is a tree and it has these branches, and etc., etc. We have to go with different ways of identifying microbes simply because they're so small and there's not a whole lot that we can see. Now on to our next big topic of discussion, methods of classifying and identifying microbes. This is where we hit the new stuff. We're going to talk about identifying them by morphological characteristics, differential staining, of which you've done some, biochemical tests, of which you will be doing some, serology, DNA fingerprinting, DNA base composition, polymerase chain reaction, or PCR, and DNA chips. One of the first ways that we started identifying microbes is by their shape, morphological characteristics, whether they were bacilli, cocci, where the flagella are if they had them, how many they had. Okay, here's an example of, I have a bacillus that's paratrichus. Now, as you've probably learned from lab, this doesn't tell you a whole lot. So we had to come up with other ways other than just shape, characteristics, to identify microbes. But it's a good way to start. We have a tendency to separate microbes into bacilli, cocci, vibrio, etc. The next thing that microbiologists came up with in identifying microbes is differential staining. Now you're familiar with the Gram stain. That was one of the first differential stains, developed by Hans Christian Gram, and as we know now, Gram staining shows us a difference in cell wall composition. Can't see it with the naked eye. They didn't have electron micrographs yet, but we can tell from this staining procedure whether it has a gram-negative type cell wall or gram-positive type cell wall. Now another one that we've come up with is acid-fast staining. This shows whether an organism has a high quantity of mycolic acid in its cell wall. Now this becomes important for a subset of gram-positive cells, the acid-fast bacteria, some of which produce antibiotics, but the ones that we're generally staining for, like in the example here, may produce disease such as leprosy or tuberculosis. Another differential stain is the endospore stain. Endospores, as you may remember from previous chapters, are the resting stage of some forms of bacteria. We use this as a way of identifying these microbes because not very many of the species of microbes make endospores. Now, if we just go with gram staining or simple staining, endospores don't stain very well. So we have to use a special stain procedure to stain these endospores so that we can tell the difference between the endospores and, say, lipid granules and other such things. Okay, all of these are staining techniques that tell us something about the composition or structure of the cell. Staining is all well and good, but we also use biochemical tests because that tells us what kind of enzymes the organism has. So this particular picture shows us a catalase test. Catalase is an enzyme that takes hydrogen peroxide, breaks it down into water and oxygen. So when you put a microbe into hydrogen peroxide, if it has catalase, then it bubbles. If it doesn't produce catalase, then it remains inert. Now the question that you may be asking is, why would the organism produce such an enzyme? Well, part of aerobic respiration produces peroxide as a byproduct. It's part of the electron transport chain. So this tells us something about the microbe. Okay, so we can do tests for enzymes one at a time. In fact, we used to do that. Nowadays, we have a tendency to combine it into what we call multi-tests, where we have in one test, we have media for different types of tests. Like here we've got a media that the only carbohydrate source is glucose. Here we've got one that the only carbon source is lysine, etc., etc. We inoculate these all at the same time with one microbe, and we determine from the results of all these tests that are done at the same time, and we're able to determine the species, genus and species of an organism. In fact, this is an anaerobe tube test. You're going to be doing this in lab. Now, it used to be, once we characterized an organism, a bacterial species, by its shape, other morphological characteristics such as cell wall composition, whether it had flagella, whether it produced endospores, then moved on to biochemical tests. We found out what enzymes it had, how it acted when it was given glucose to eat, etc. Then we could develop serological tests for it. Serology basically is the study and use of antibodies. Your immune system produces very specific proteins that attach to other very specific proteins. The ones that your body produces we call antibodies. What the antibodies attack are called antigens. So once we have characterized an organism, we can take specific proteins from that organism and inject it into, say, a mouse or a chicken, have it produce antibodies, and we can attach a stain to it, and you get fluorescent staining. Okay, this is serology, using antibodies to help us identify organisms. You can only do this if you already have the organism characterized and you have it growing in pure culture. Now, this is fluorescent staining. We talked about this in chapter 3. We've also developed a test called the ELISA test. Stands for enzyme-linked immunosorbent test. And basically what we do is we make a sandwich out of an antibody or an antigen to amplify what we see so that we can see whether we have that antigen or not in the little wells. We'll talk about this more when we talk about immunological testing. But I wanted to introduce the concept of using antibodies to identify microbes, or to identify if a patient has produced antibodies against a certain microbe. Now, serology sped up identifying significantly. Once we were able to develop what's called monoclonal antibodies, antibodies that we know are against just one antigen, this sped up identification significantly. Now, all of these things that I've been talking about are still heavily used, but enter the brave new world of DNA identification. This has changed identification significantly. Let's talk about DNA fingerprinting and DNA base composition. There are enzymes called restriction enzymes that cut up DNA at certain spots. And if you take a DNA sample and you put restriction enzymes in and cut them up at different spots, you get different sizes of pieces of DNA depending upon the organism. Okay, and so you put them onto a gel. It's kind of like agar, but it's a little bit different. And you put an electrical current across. You put a positive charge here, you have a negative charge here. Which way is the negatively charged DNA going to go? It's going to go toward the positive electrode. And we use this to separate out the pieces of DNA by size. The smaller pieces wiggling through the holes in the gel faster than the bigger ones. Now, each lane, we cut a little hole here, we call it a well, and we put one sample here that's been chopped up. We put a different sample here that's been chopped up, etc., etc. And we generally run a control that has known sizes of DNA. And when these separate out, they make a different pattern for each sample. So, for example, for these wells, they all had DNA that's about this size. They also had DNA pieces that are about this size. This one had more. This one has a band here, then a band here, then these guys share, but these guys don't have that band, etc., etc. It's kind of like a fingerprint. It's a way of identifying the unique characteristic of a microbe based on something other than eye color, height, and hair color, since microbes don't have that. So these pieces of DNA are kind of like a fingerprint. They're unique for the bacterial species. So we would use this for identification. This is before we were able to do sequencing. Okay. Now, also before we were able to do sequencing, which is basically determining the nucleotide sequence, the order of adenine, thymine, guanine, cytosine that's unique to the organism, that's unique to the chromosome, we also did DNA base composition. We would basically take the DNA, chop it up into the individual nucleotides, and see how many G's and C's we had. If we knew how many G's and C's we had, we knew how many A and T's we had, and funny thing, sometimes this is characteristic for different groups of organisms. That's as good as we could get. With the advent of more technology, we can now determine the actual DNA sequence and look to see how related it is to of one bacteria to another bacterium. And this changed things around. We had to move different microbes around because how we were grouping them before, and it was the best that we could do, was kind of like taking a room full of humans and saying, okay, all the blondes move over to the right, you're all related. All of the folks that are brunette move over to the other side. Now we're going to separate you by whether you like Italian food or whether you're allergic to milk. And as you know, that doesn't necessarily indicate relatedness, but it was the best that we had. Now that we can actually do DNA tests, it's kind of like taking that same room full of people and saying, okay, everybody who are first cousins, step forward. Okay, all of you who are second cousins, okay, you're going to be in the second tier, and so on. So this has advanced identification and classifying of microbes because now we can see who's actually related to whom, whereas before we had to just separate them on whether they liked milk or not. Now, to do all of this DNA testing, you've got to have a lot of DNA. Unfortunately, or fortunately, in a cell there's not a whole lot of DNA. The cell doesn't need a whole lot, so there's not a whole lot there. So we had to come up with a way of making a whole lot of copies of the DNA so that we had enough to see. Think of it as, in lab, getting enough bacteria to make a colony. You can't see with your naked eye one bacterium. It's just too small. But if you multiply it to about 1 million organisms and put them all in the same spot on agar, we can see that colony. Well, we do kind of the same thing with DNA. And we do it by means of polymerase chain reaction, or PCR. So remember, a polymerase is an enzyme that makes a polymer. In this case, we use DNA polymerases. And we basically go through replication or duplication of DNA outside of the cell. So here's how it works. You take your sample. It's double-stranded sample, and we put it into a container with primers. Remember that DNA polymerase needs to have an RNA primer, and we put it in with a whole bunch of nucleotides, free nucleotides, and what we do is we heat it up to unwind and separate the strands of the DNA. Now in the cell, helicase would do this job, but in a PCR thermocycler, heat does it. That's where the thermo and thermocycler comes from. So we heat it up enough to separate the two strands, but not enough to start breaking down the nucleotides from each other, and we put in a DNA polymerase. The DNA polymerase makes a copy of each strand of DNA. So from one double-stranded DNA, we get 2 double-stranded DNAs. Then we do it again. We heat up these strands, separate them, we have more primers, we have more nucleotides. The DNA polymerase, which we isolated from a thermophile, so it's not denatured by heating up the DNA, okay, and it makes another copy. So from these 2 we get 4, and etc., until we keep going through these cycles We make a chain reaction and we make a whole bunch of exact copies of our original sample. Now, PCR is used in more than just microbial identification, making enough copies of our samples so that we can take a look at it through various DNA techniques such as DNA fingerprinting. Also, we use it in identifying human bodies, identifying where the blood came from at a crime scene, identifying which suspect did the dirty deed, who's the dad, all sorts of good stuff. We can use PCR to amplify any DNA that you're talking about. Generally, we do this before we do other techniques. So, going back to DNA fingerprinting, if I put just the DNA from one bacterium on here and tried to run it, it would be too faint for me to be able to see. But if I take the DNA from one bacterium and I put it through PCR and I get millions of copies of that DNA from that one organism, then I can see it with my naked eye when I run the gel. Now here's another example of using amplified DNA, making lots of copies of DNA and applying a new technique to it. Somebody got the smart idea of making basically a computer chip to identify bacteria, to identify different genes even. What they did is they took a silicon chip, just like what you would put a computer chip on, and instead of putting little wires on-off circuits, they took little probes, little pieces of DNA from different microbes they wanted to identify, and they put them on little spots. So what we see here is little spots. And we put enough of these probes, okay, little DNA sequences that we're going to look for in a sample, all over on this chip. This is basically a multi-test, but for DNA instead of looking for enzymes. So then you take your sample, you run it through the PCR, you attach a fluorescent stain to it so that that fluorescent dye lights up when that single-stranded DNA becomes double-stranded. Now, double-stranded DNA is looking for its opposite number to hold hands with and become double-stranded with. So when you run your sample through, if it finds its match, it grabs a hold and lights up. Now here we can see that not very many strands found their buddy, their opposite number. Here we found more. Red is not so much, orange, yellow more, green is the most. So using this pattern, which by the way computer reads, you can say, oh, this is organism X. Ah, okay. Or even better, because identifying just one kind of organism would be kind of a waste of the ability of these microarrays, or what they're called. The book calls them DNA chips. We can take a whole soil sample that has hundreds of bacterial species, hundreds of fungal species, hundreds of eukaryotic species, hundreds of archaic species, and we can amplify the DNA, run it across a chip, and we can identify which species are there. Okay, so that's what they're doing now. They're not identifying individual organisms. We can make in the lab the DNA sequences that we're looking for, that are characteristic for that organism, that are going to catch the entire genome. Actually, we're looking for parts of genomes, but we'll think of it as catching the entire genome. And we can screen an entire sample, and we can not only screen an entire sample, but we can pick up bacteria that we haven't been able to grow in the lab from their DNA, from catching them on these microarrays, on these DNA chips. We can see bacterial species that we haven't been able to grow, that are too fastidious for us to grow in lab. Using this type of technique, we've found that 90% of the bacterial species, unique bacterial species we've been able to identify this way, are unknown to us in the lab. We're not able to grow them. We're finding this when we do studies of DNA of samples from the soil, from ocean samples, from the human gut biome, from periodontal pockets, you name it. Most of those microbes we can't grow, but we can identify them by their DNA. Alright, that's it for this chapter. As you know, the first part of it was a review of what we covered before. It's always good to hit it again. And also just hitting the tests that are used most frequently right now. The book talks about some other ways of identifying microbes, lipid analysis and different things. Those aren't being used quite as frequently, and you're probably not going to run into them as much as you are the ones that we hit. So those are the ones that I want you to know. So make sure that you go back and take a look at them. This test is probably going to be a little bit easier because we're going to be doing some of these ID tests in lab. And as always, feel free to listen to this as frequently as you would like.